I would like to welcome our guest today and tell you a little bit more about our special guest today. Professor Séverine Morisset-Lopez is a lead researcher at the French National Center for Scientific Research, so in short CNRS, based at the Center of Molecular Biophysics in Orleans in France. She obtained her PhD in molecular pharmacology from the University in Paris, after which she joined the Center of Psychiatry and Neurosciences, where she began her career in research. So I would like to stress out that over the years, Professor Morisset-Lopez has developed a really strong expertise in molecular and cellular neuropharmacology, with a particular focus on G protein-coupled receptors, GPCRs in short. And I must tell you that it's very valuable in drug discovery, especially today, especially in the context of functional selectivity. So it's really great if you really pay attention to it. Professor Morisset-Lopez research primarily explores the role of GPCRs, obviously, neuronal signaling and their implications in different neurological disorders. And she employs advanced techniques such as BRET to study receptor interactions and signaling pathways in both neuronal and non-neuronal cells. Which is, as I mentioned, very, very important for drug discovery nowadays. I would also like to say a little bit about the fact that Professor Morisset-Lopez is the author of many articles and also a patent, which is very exciting. It's not easy to be an author of a patent, and this reflects her extensive contributions to the field. She is also very actively involved in international collaborations, including us as well. I really hope that we will publish our paper soon with the results from Professor Morisset-Lopez lab. And she really contributes a lot of valuable knowledge to different interdisciplinary projects that bridge neuropharmacology and molecular biology. So I'm very, very excited that Professor Morisset-Lopez agreed to deliver a lecture for you, and she's here with us. And I now give the floor to Séverine to have a lecture. Thank you very much.
Thank you so much. Thank you so much, Claudia and Carolina, for this introduction. Especially Carolina, yes, we collaborate, so it's a nice pleasure to be there in front of you, and today, yes, and I would like first to thank you, the organizer, to give me the opportunity to present our works and this technique. And so I will share my screen. Hope it will work.
Yes. Let's try. If I can add one more technical thing. Students, if any questions come to your mind, please write them in chat. And after the presentation, we can discuss with expert Professor Morisset-Lopez. And so please, if something comes to your mind, if something is not very clear to you, write it in chat. Thank you.
Okay. Can you see my screen?
We can see the screen, but not in an editing mode, not the presenting mode. Oh, now the presenting mode.
And the presenting mode. Okay. So I will try to put the pointer. Okay. So can you see the pointer? Yes. Okay.
Yes, we can.
Yes, you can. Okay. So yes, it's a real pleasure today because it's an opportunity for me to present the bioluminescence resonance energy transfer as a powerful technique in neuropharmacology, especially for discovering new ligands of G protein coupled receptors. So during this talk, first I will present you the technique, the advantages, the drawback of the technique, and then I will present new results that we obtain in my team. So my team is entitled Neurobiology of Receptor and RNA for Therapeutic Innovation. And our main topic is on G protein coupled receptors. So, yes. So, in fact, our strategy of research is based on a multi-scale analysis. So first, we develop new screening-based assays. Most of them are based on the BRET technology to identify new therapeutics, such as small molecules, but also antibodies targeting our receptor of interest. Then we evaluate the pharmacological property of the new ligand that we identify on various cell-based assays. So we use different cell lines as well as primary neuronal culture. So the next step is to evaluate the effect of this new molecule on various animal models to evaluate the therapeutic potential of this new ligand. Then we evaluate the expression of some biomarkers by PCR analysis or histological studies. Okay. So as I told you, my main research is based on the study on the G protein coupled receptors, so which represents a large family of receptors comprising around 800 members in human. They are involved in many diseases, and there have been over 30% of approved drugs that targets these receptors. So during the last decade, I wanted to tell you that we have seen an evolution in pharmacological concepts. In fact, we know now that the receptor can be modulated at different levels. First, they can be modulated by orthosteric ligands, but also by allosteric ligands, which act at a different site. They can also be modulated by their interaction with different protein partners, and such as G protein, but intracellular proteins such as aristin to modulate the signal transduction. I would like also to say that this receptor also display basal constitutive activity, meaning that this receptor can adopt an active conformation and trigger intracellular signaling even when no ligand is bound. So this basal activity is can be also modulated and blocked by some molecules that we name inverse agonists. So, and now it is well known that this receptor can form dimers or higher oligomers. So, this GPCR can be modulated by different ligands, chemical molecule, biogenic amide, peptide, and antibodies. And in drug discovery, after identifying a molecule that binds to a receptor, we need to define its pharmacological profile. I mean that, for example, an agonist is a molecule that binds to the receptor, activates it, and produces a biological response. An antagonist also binds to the receptor but doesn't activate it. It blocks the receptor and prevents other molecules like agonists from binding and triggering a response. An inverse agonist binds to the receptor and is able to reduce its basal activity, producing an opposite effect that of an agonist. So these three types of ligands help us understand how drugs can either stimulate, block, or suppress receptor activity. So agonists are useful when you want to mimic the natural ligand. Antagonists are used to inhibit overactive signaling, and inverse agonists are useful when receptors have constitutive activity. They are acting even without a ligand. So understanding this mechanism is a key for designing drug and which precisely control the activity of the receptor. But I want to go further and to introduce you another and relatively new concept in pharmacology, the concept of bias signaling. This concept of bias signaling has been introduced by Professor Lefkowitz and Kobilka, who received the Nobel Prize in 2012 for their research. And they propose that in contrast to classical ligands, which turn on or turn off the downstream signaling pathway of a receptor, there are some bias ligands which are able to selectively activate one signaling pathway while reducing activation of other pathway. And this kind of ligand, by reducing activation of the pathway, could be responsible which could be responsible of adverse side effects, are supposed to have higher therapeutic potential compared to classical ligands. So for any given receptor, really it's essential to characterize and classify drugs based on their ability to activate different signaling pathways. So this means that we must evaluate how a single molecule could affect multiple signaling pathways and not just one. So, to define the pharmacological profile of a ligand, it's now more complex because we have to evaluate the effect of one ligand on different signaling pathways. And in this talk, I will show you how BRET, the bioluminescence resonance energy transfer, is very useful and can help to achieve this purpose. So BRET, you know, because you answer well during the quiz, so you know that bioluminescence resonance transfer originates from a natural bioluminescence system found in marine organisms. So here you can see here that in jellyfish, named Icora victoria, this animal in fact produces light through a bioluminescent reaction. So in fact, in this yellowfish, there is a bioluminescence protein named icorin, which induces bioluminescence, and the blue light can be transferred by resonance energy transfer to green fluorescent protein that we could find also in the sea, resulting in a green fluorescence. So this natural process is a biological example of resonance energy transfer. I want to show you another example. Sorry, oops. Okay. Another example. So in the sea pansy, they isolate an enzyme, the Renilla reniformis. Is another bioluminescent protein. And in this sea pansy, you could also detect BRET resonance energy transfer as soon as a green fluorescent protein is closer to this bioluminescent protein. So, it was very interesting to see that in science we use a natural phenomenon. But what is exactly BRET resonance energy transfer? So this method is a proximity-based assay that could detect protein-protein interaction, also conformational changes via a non-radiative energy transfer from a bioluminescent donor to a fluorescent acceptor. And so the energy donor and acceptor are, so in science, to detect protein-protein interaction, we need to fuse the protein of our interest, for example, the protein X. We need to fuse this protein to the bioluminescence protein Renilla luciferase. And the other protein that we want to test, the protein Y, is fused to a fluorescent protein as acceptor. So, when you have the fused protein, after addition of the substrate of the bioluminescence donor, the Renilla luciferase could emit light that excites the acceptor fluorophore, but they need to be very close, within 10 nanometers. So indicating the close proximity between the donor and the acceptor. This is the principle of this technology, but I wanted to tell you that in fact BRET was first inspired by FRET. FRET is fluorescence resonance energy transfer. But in fact, first, BRET has been developed, in fact, to overcome FRET limitations. So I will show you the difference between BRET and FRET. So as you can see in this table, first of all, so BRET relies on a bioluminescent enzyme like luciferase to generate light, unlike FRET, which needs an external excitation light. So due to the excitation light that is required for FRET experiment, this induces a high background of autofluorescence. So BRET has the advantages to have low background noise and no autofluorescence or photobleaching. In addition, due to the excitation of the donor, this can induce phototoxicity and require precaution when you perform FRET experiments. In comparison, you can perform BRET in real time in live cells and to follow the dynamic of the interaction between protein. Another advantage of BRET is that we use only a microplate reader to read the luminescence with a specific emission filter, whereas BRET requires a more complex system, in particular fluorescence microscopy or confocal. In addition, BRET, there is a high spectral separation. So really, we could see the interaction between two proteins, even if the signal is low. So BRET is very effective, and we could detect rare interaction. In addition, BRET is, we could perform high throughput screening by BRET, and it is, we can do it with by FRET, but it's a little bit more complicated. So why and when we can use BRET methods? As I told you, my main topic is on the G protein coupled receptor. So BRET is a very useful method first for drug discovery, targeting GPCR. So we can perform by BRET high throughput screening. So for example, you have to fuse your receptor of interest with a bioluminescence protein. If you want to follow the downstream signaling involved in the activation of the receptor, for example, beta-arrestin, you can fuse beta-arrestin to a fluorescent protein, and then after addition of ligand or addition of different compounds coming from a library, you could detect which compound will be able to activate the receptor. So it's a way to perform screening assays. In addition, with BRET, you can monitor the interaction of protein-protein in real time. So, for example, you can follow in real time the interaction between the receptor and the G protein, between the receptor and arrestin, so you can follow beta-arrestin recruitment after a receptor activation. You can follow receptor internalization, and you can also measure by using BRET biosensor specific downstream messenger involved in the transduction of the receptor. For example, you can measure cyclic AMP levels. BRET is also very useful when you want to study dimerization or oligomerization between two or more receptors subionic. So BRET is very useful to monitor in real time dynamic biological processes in live cells. So I suggest you to read our review because we wrote a review a few years ago on this technique and how we can apply this technique to study GPCR. So now we can we can go to the in more detail in the use of this technique. So first of all, to perform BRET experiments, you need to take into account several factors which can influence resonance energy transfer. First, you need to use a compatible couple. I mean the energy donor emission wavelength here in blue has to overlap the energy acceptor excitation in order to gain energy transfer. So this is the key point to select the donor and acceptor when you want to perform BRET analysis. In addition, there are also several other factors which influence the efficiency of BRET. The distance between the donor and acceptor, the orientation of the donor and acceptor, and the ratio of the donor and acceptor. So first, as you know, energy transfer between two proteins only occurs when donor and acceptor protein are within the 1 to 10 nanometers of each other's. So BRET is very sensitive to detect very close interaction. Second, another parameter that you have to take account is the orientation of the donor and acceptor, which could also affect the efficiency of the signal, even at a short distance. If the orientation of the donor and acceptor is not good, you will not detect any transfer. What is really very, very important is the ratio of the donor and acceptor. So you need to be careful because the expression ratio of the donor and acceptor protein must be very and carefully controlled, because too much acceptor can increase the background and the non-specific signals, while too little reduce the sensitivity of the technique. So you need to confirm that when you get a BRET signal, you have to be sure that the signal is due to specific interaction. So in fact, we perform BRET saturation curve assay to evaluate specific interaction. So in this experiment, the donor levels is kept constant here in blue, and we increase the amount of the acceptors. And we got an hyperbolic curve when we obtain specific interaction. And we reach, in fact, a plateau. And this plateau is due appear when, in fact, here you can see that when all the donor interact with an acceptor, you reach the maximum signal that you can obtain. But in the case of non-specific interaction, you observe an increase of the signal that is linear. So it is really important to perform different kinds of experiments to evaluate the specificity of the signal. So now I want to introduce, in fact, the different BRET system, because over the past years, different BRET systems have been developed. So the first BRET system was described in 1999 using Renilla luciferase as the donor, and YFP, the yellow fluorescent protein, has acceptor. So when you use the substrate of luciferase, you get a blue emission light at 418 nanometer. And when you study interaction with a yellow fluorescent protein, if there is a resonance energy transfer, you can observe an increase of the signal at 530 nanometer. So this BRET system is very simple, is widely used, and it could be used in a physiological expression. You can express your protein of of interest at very low level. You can perform studies in the primary cell culture. So this BRET is very used in fundamental research and for screening. There is another BRET system. In this case, so they develop, in fact, the problem of the first one, I could show you again. I will go back. The problem in this system of the BRET1 is that the difference of the wavelengths between the emission of the donor and acceptor are relatively close. So you can have a background due to the emission of the donor. So in the BRET2 system, they try to increase the distance between the emission of the donor and acceptor. So they develop a new substrate named Deep Blue C. So this substrate used with renilla luciferase induce an emission peak at around 400 nanometer. So you can use also as acceptor the GFP, and you can evaluate the interaction of two proteins between these substrates that have been developed, the Deep Blue C substrate for the BRET2 system. So what is the advantage? Better spectral separation. But the problem is that, in fact, the Deep Blue C doesn't induce a very bright luminescence, very bright signal. So we have to increase the amount of the donor. We have really to overexpress the donor. So it could be a disadvantage because we are not in the physiological expression of proteins. So it's this technique is not very used for certain. Is not used when you want to evaluate the interaction at physiological level. However, you can use BRET technology, BRET2 technology for fundamental research, screening, and also for cell imaging. Another system is the BRET3. So when so BRET3 has been developed how can I say that, to have a redshift and to detect interaction in more redshift wavelengths. This could be used because, and very useful when you perform experiment in living cells, because in living cells there is an autofluorescence background in the green wavelengths. So that's why they develop this kind of BRET to have less background. So to develop this system, so they develop an acceptor protein. So the acceptor is named M orange or DS red. They also develop different substrates, so vivirin or luciferin, sorry. So as I told you, the main advantage of this technique is the reduced autofluorescence in tissue in particular, but rarely it's less common and not really used in a GPCR field. But you could apply for screening if you want. So now I will go to the nano-BRET system. So nano-BRET is the latest and the most advanced system. In NanoBRET, you could see by the name, we use a nanoluciferase. The nanoluciferase is a very small and bright luciferase, which is used as a donor. So nanoluciferase was developed by Promega, a company who in fact isolated this luciferase from a deep sea shrimp called Oplophorus gracilirostris. And in fact, they isolate this luciferase, but it's a huge and large protein, over 100 kilodalton. So they improve the enzyme, and they try to find the minimal part of the protein that induce the bioluminescence. So in fact, they isolate the subunit responsible for the catalytic activity of the enzyme. And this luciferase, nanoluciferase, is small, around 19 kilodalton. So Promega improved also the system by developing a new substrate named furimazine, and this substrate is it costs a lot, in fact, but it gives a lot of results, because we get a very strong signal when we use this kind of substrate. I wanted to show you in the next slide why it's very why nanoluciferase is very useful now to develop new BRET system. So as I told you, nanoluciferase is very small compared to the other luciferase, only 19 molecule, she had 9 kilodalton compared to Renilla luciferase or Firefly luciferase, which are a bigger protein. So what is important is that when you fuse your protein of interest with a bioluminescence protein, you have to verify that the fusion of the bioluminescence protein doesn't modify the activity of the protein that you want to follow. So when you use a smaller bioluminescent protein, it could be useful because you avoid or you favor minimal steric hindrance. So it's the size of the nanoluciferase is very useful, and now all the BRET that we develop now, we use this small bioluminescence protein. So I wanted to show you the different experiments performed by Promega to demonstrate how nanoluciferase is bright. So they compare the activity of nanoluciferase with the other luciferase, Fireflies and Renilla. And you could see in this figure, in this graph, that the nanoluciferase induce 100 times more increase of luminescence compared to the other bioluminescence protein. When they perform these experiments on purified enzymes, or when they express the different enzymes in mammalian cells, so they could detect a very strong signal after expression of this nanoluciferase. So nanoluciferase has excellent physical property because it's stable at different temperature. It is active over a broad pH range. It's a monomeric enzyme, so we could use as a reporter. There is no post-translational modification, no disulfide bonds. So this support that you could express the protein inside the cells, and the activity of this protein is still intact even in a specific condition. You could see here that when you express this nanoluciferase in cell, you have a distribution in the cytoplasm. So there is no apparent compartmental bias due to the expression of this nanoluciferase. So Promega also confirmed that this nanoluciferase is very useful when you perform and when you want to set up a screening-based assay. In fact, they compare screening assays performed with nanoluciferase and firefly luciferase, and they calculate the Z prime factor. I don't know if you are familiar with this factor. So Z prime factor is a statistical parameter that evaluates the quality and the reliability of a high throughput screening assay. So it takes into account the dynamic range and the variability of both the positive and negative control. So this factor is close to 1, it is the best. So you could see that in this case, so the Z prime is very high compared to those obtained with the firefly luciferase. So here is one of the application of BRET to perform a screening assay. So here they used a large library of compounds. They tested at least 1,000 of compounds to detect. The aim was to detect some inhibitors. So you can see one of the application of BRET methods. So to conclude on the BRET and nanobret in particular, so in nanobret, you use as a donor the nanoluciferase, as acceptor the YFP, as a substrate the furimazine, and you detect the emission light at 460 nanometer and 530 nanometer. So the main advantage of nanobret is because it's 100 brighter than vanilla luciferase, so extremely bright, and you have a stable emission, excellent sensitivity, and the signal to noise ratio is very good. Due to the small size of nanoluciferase, this minimizes steric hindrance. This nanobret is very useful to perform kinetics in live cells. So you could apply this nanobret to monitor protein-protein interaction, to monitor in real time the formation or dissociation of some complex, to study the recruitment of beta-arrestin, or to perform drug screening and binding kinetic assays. Here I summarize in this slide the different BRET systems that I present you. So you have the different substrate, the wavelengths of the donor and acceptor. Yes, it's the summary of the technique. So now, what about the advantages of BRET? So as I told you, compared to FRET, there is no external excitation light needed. So this induce that you have low phototoxicity, no photobleaching. You have a high signal-to-noise ratio, and especially with nano-BRET. Oh, sorry, there is something disappear, but it minimizes autofluorescence. So especially when you use the BRET3 system, which involve redshift dyes. And so the BRET is very useful for screening assays. But as all technique, there are some disadvantages because it requires luciferase substrate, and in some cases they can be very expensive. And another point, if you want to perform kinetics over the time, the signal intensity may decrease over the time. And what is very crucial, and I told you before, you have to optimize the ratio between the donor and acceptor. And another disadvantage, you need to perform protein fusion. So you need to fusion your protein of interest with a donor and acceptor. So by fusion of this protein to the donor and acceptor, you could modify the functional property of your protein of interest. So you have to perform some controls. So some system may require specific labeling, and you need to have a specific equipment to detect and to measure luminescence. So now how to perform BRET in practice. So first, as I told you, you have prepared the donor and acceptor. So you have prepared plasmid, and to fuse the sequence of your protein of interest with the sequence of the bioluminescence protein or the fluorescence protein. So first you have to perform molecular biology. Then as soon as you have your plasmid, you use cells. In our case, we use egg cells, which are commonly used in pharmacology. So you cultivate the cell in 10 centimeter plate, for example, or in a 6-well plates. So you have to transfect the cells with your plasmid. So for transfection, you can use lipofectamine or calcium phosphate, depending on what you want to do. If you want to have a high expression, or if you use primary cell line, you need to use lipofectamine. So after transfection, you keep the cell at 35 degrees for 24 or 84 hours. And then 2 days, most of the time, 2 days after transfection, you collect the cells. So you eliminate the medium, you wash the cells with PBS, and you detach the cells by pipetting or by using dissociation reagents such as Versane or trypsin, for example. Then you resuspended the cells with a buffer, saline buffer. And then you can distribute the cells in 96 well plates at 50,000 or 90,000 cells by well. Then, as soon as you have distributed the cells, you can prepare the ligands that you want to test. So you prepare the ligands, and if you want to detect if a ligand as soon as interacts the receptor, it induces the recruitment of different protein partner. So you had the ligands that you want to test or to screen. So then you had the ligand, you incubate the ligand for appropriate time, for example, between 5 or 30 minutes. Then you add the substrate. You can use, depending on the donor and the donor you use, you can use selanterazine, furimazine, so depending on the donor. So then you read luminescence immediately and you use different filter to detect the luminescence at the different wavelengths at 460 or 530 nanometer. Then you have to calculate BRET ratio. So I will show you how to calculate BRET ratio just after. Just this slide to sum up the materials that you need. So the plasmid, the cells, the culture medium, transfection reagent, the substrate, the different ligand. You need a white 96 well plate or different well plate, but white because to decrease the background signal. And you need a plate reader equipped with filter so depending on the system, the BRET system that you use, you will choose a different filter. So you also need basic equipment, cell culture wood, an incubator, micropipette, the plate, and if you want to check if you want to check the transfection efficiency, you have to use a fluorescent microscope to verify the expression of your acceptor. So here is an example. So to detect, for example, the interaction of GPCR with beta-arrestin, so you need to do the protocols that I described before, and then you have to use a microplate reader. So typically in this microplate reader, you have a photomultiplier tube, the PMT, which ensures accurate measurement of the donor and acceptor emission and allowing you to calculate the BRET ratio. So the apparatus, in fact, detects, so it detects in a specific well, for example here, it can detect the emission of the donor, then the yield shuttle, and then to detect the emission of the acceptor. But you could have different microplate reader. Some of them have two PMT, so you can detect in the same time the emission of the donor and the acceptor. So in my team, we use this microplate reader, Berthold, from Berthold, but you have here different microplate readers. So here is a few words to tell you how we calculate the energy transfer. So you have to calculate a BRET ratio. So this BRET ratio is calculated, in fact, by dividing the acceptor signal obtained, for example, at 530 nanometer by the donor signal obtained at 480 nanometer. So this ratio reflects the proximity of the two fusion proteins and is proportional to the interaction. So we need to take into account of the background BRET ratio. I mean, when you have no interaction, you still detect a signal, a BRET ratio, because you have an emission at this wavelength at 530 nanometer and at the wavelength due to the emission of the donor, the peak of the emission of the donor. So when you express only the donor, you calculate background BRET ratio and to evaluate the energy transfer between the donor and acceptor, you have to do this calculation. So you have to use the BRET ratio obtained here and to subtract the background BRET ratio obtained only with the donor or in controlled condition when you do not use a stimulate ligand, so when you use buffer. And then you multiply by 1000 and then you obtain a ratio in millibret. So just some tips when you perform when you want to perform BRET experiments. So as I told you, you have to ensure that nanoluciferase or your bioluminescence protein does not affect the receptors pharmacology. You have to maintain consistent expressions levels to avoid saturation or insufficient signal. So you have to optimize the ratio between the donor and acceptor. Use white plates to reduce signal crosstalk and to improve detection sensitivity. You have to protect the plate from light during detection to minimize signal degradation. And always include negative controls, no ligand, donor only or acceptor only for accurate interpretation. So here is a video. I hope that I will able to start the video. Perhaps I couldn't start the video. I will try again. Yes. So as you can see here, the BRET is a bioluminescence resonance, a natural phenomenon. So in the team, yes, we perform culture in 6-well plates. So we did transfection in 6-well plate. We first evaluate whether the transfection did not affect the cells if they are healthy. Then we evaluate whether the expression of the acceptor is good. So we use this fluorescent microscope to detect the fluorescence of the acceptor. So as soon as we know that the transfection was good, so we start to perform BRET experiments. So as you could see here, we can see the cells attached on the plastic. So then we detach the cells by pipetting, as you can see here. So you can see here that we detach the cells, we resuspended the cells in the Heinz buffer. Then we distribute the cells, and we had the different ligand in the 96-well plates. And finally, we read the BRET signal in this microplate reader. So first of all, we first check that we got a signal to detect whether the donor is allowed to detect a luminescent signal. So here, yes, sorry, I had the celanterasine now. So, and then we measure the signal at 2 different wavelengths. So you have 2 different, here, 2 different measure at 430 nanometer and 480 and 530 nanometer, and we calculate a ratio. So, yes, so here is, sorry, I will stop the video. So I'm not sure if I will have the time to show you how in the team, because I plan to show you how in the team we use BRET. I don't know if I will have the time.
Sure, go ahead.
Yeah, okay. So now I want to show you how we use BRET on our favorite receptor, the serotonin 5-HT7 receptor. And I will show you how we perform screening and how we evaluate the new ligand in different cells assay as well as in vivo. So how we perform a multi-scale analysis to better understand the activation of a receptor. As you can see here, the receptor is highly expressed in brain with higher expression in thalamus, hippocampus, and cerebral cortex. So the receptor is also expressed in peripheral tissues. It is positively coupled to G protein and to adenylate cyclase. It activates adenylate cyclate, and as a result, we observe an increase of cyclic AMP level. Then the receptor triggers ERK activation and ERK phosphorylation. So in addition, the group of Professor Ponymaskin showed that the receptor is also coupled to G-alpha-12 proteins, which allow the activation of mTOR and ROA pathways, which is important for synaptic plasticity and neuroid outgrowth. Additionally, I want to tell you that the receptor exists in multiple isoforms, which differ in the length of the C-terminal loop of the receptor. However, to date, no difference in their pharmacology profile have yet been described, and the role of these different isoforms remain largely unknown. So its pathophysiological role has been evaluated thanks to the identification of various ligands. So therefore, numerous agonists have been developed, such as ACE19, the Leopoldo compound, LP44, or the 5-CT. And these agonists are able to activate the receptor and have shown to have beneficial effects for the treatment of cognitive disorders, headache, and for the treatment of pain behavior. So in addition, there are also several antagonists of this receptor that have been identified. And I will show you, because we use this compound, the SB269970, in our experiments. So it has been demonstrated that these agonists have beneficial effect on depression, stress, anxiety, and schizophrenia. So therefore, this receptor is considered as a very, very interesting target, as evidenced by the number of patented molecules and the interest of a number of biopharmaceutical companies. So today I will focus my talk on the role of the receptor on pain, because there are several data in the literature showing and demonstrating the role of the receptor in pain. In particular, the receptor is expressed in different regions involved in pain circuit. So it is expressed in dorsal root ganglia cells, in the dorsal horn of spinal cord. It is also expressed in several supraspinal area here, as you can see here. And in addition, several studies have been performed demonstrating that the activation of the receptor reduces pain. So, for example, I show you here data obtained by the Professor Brenshaw, who demonstrated the beneficial effect of agonist of the receptor to control pain. So they use an inflammatory pain model. So after injection of capsaicin in the paw of the animal, you could observe an hypersensitivity of the animal here. And when they injected increasing dosage of an agonist of the receptor, this molecule in black, they could reverse this hypersensitivity. And meaning that the receptor is important to control pain behavior. And in another test, they confirmed that the effect of this ligand, but other ligands, is due to its binding to the 5-HT7 receptor, because here in another test, the formalin test, so they use another ligand, the E55888. So when they administrate the ligand in the animal, they observe a decrease in the paw licking time. But when they did the same experiments in knockout 5-HT7 knockout mice, they do not observe the effect of the ligand, demonstrating that the receptor, the activation of the receptor, is really important to control pain. However, when I started this project on 5-HT7 receptor, we identified several challenges to study the receptor. In fact, agonists are not very selective. They bind to the receptor, but they also bind to other serotonin receptor. Some of ligands have low biodisponibility. They do not enter very easily in the brain, so it's a limit when you want to develop new molecule and new therapeutics in neuroscience. There is no specific ligands for isoform and no specific antibodies. So in the team, we developed two complementary strategies to identify new ligands of the receptor. So first, to identify small molecules, but also we developed recently a new program to identify antibody and especially the antibody fragments named nanobodies to modulate the activity of the receptor. But today I will not have the time to go in detail and to present you this research on the nanobody, but I will show you our results on the small molecule. First, I want to give you an example how we applied BRET to identify a new ligand of our receptor. Classically, when you want to screen ligands, in the past years, when researchers wanted to identify and characterize new ligands, they used radioligand binding assays. But this assay, there are some limits because you use radioactivity, so it's bad for the environment. It costs, and you have to take into consideration the half-life of isotopes. So that's why the BRET method has been developed a few years ago in 2015. And it's Professor Stoddart who used nanobret to identify a new ligand of G protein receptor. So nanobret has some advantages because you do not use radioligand, you use fluorescent ligand, and these assays are adaptable to high throughput screening. So I will show you how we develop in the team this nanobread-based assay. So to set up a bread-based assay, so you need to fuse your receptor to nanoluciferase, so the bioluminescent protein, and to observe an energy transfer between a donor and acceptor, we use not a protein but a fluorescent ligand. So we need to develop fluorescence ligands to detect the interaction of the ligand with the receptor. So, so you can see here the emission peak of the nanoluciferase at 460 nanometer, and this emission peak should overlap with the emission peak of the acceptor. So here we use different kind of body piece, fluorescent fluorophore named both DP, has an acceptor. So here is the workflow that we developed to set up this nanobread-based assay. So first you have to generate different constructs to fuse your receptor to nanoluciferase. So we perform different constructs because we changed we had the nanoluciferase at different position within the N-terminal loop of the receptor. Then, because we had this nanoluciferase to the receptor, we have to validate the functional property of a receptor. So we perform functional assays. Then we have a collaboration with chemists to they synthesize different ligands which differ by the nature of the fluorophore and the length, the nature and the length of the linker between the ligand which target a receptor. So we use, for example, we use a specific ligand of a receptor that is coupled to this fluorescent protein. Then we have to evaluate different combination of BRET pairs in wool living cells. So we evaluate different BRET pairs, different fluorescent ligands, and then we choose the best one. We verify the specificity of the signal by performing a BRET saturation curve. So, and I will show you just after. And this test is now available as a screening test because we could perform competition binding assay with non-fluorescent ligands. So a non-fluorescent ligand here in green will do competition with the fluorescent ligand. So as soon as one ligand bind to the receptor, it do competition, so you should observe a decrease in the BRET signal. So I will show you some of our results. So first, we evaluated here the different fluorescent ligands to identify the best one, the one that allow a higher BRET ratio. And then we have to confirm the specificity of the signal. So as I told you before, we perform a BRET saturation curve, as you could see here. So we use the fluorescent ligand in increasing concentration. So you observe a saturation signal, and this signal is very specific because when we use a competitive ligand, a non-fluorescent ligand, so SB269, here you could see that we observe a strongly decrease of BRET signal. So then we confirm that our BRET assay could be useful. So we compare the affinity that we got with our BRET assays, a nanobread assay, compare with the affinity obtained with radioligand binding for different known ligands of the 5-HT7 receptor. So for 5-CT, the reference agonist, you could see that the value are very similar, so confirming that our BRET-based assay is useful to perform screening assays. So we use this test as well as a radioligand binding assay in collaboration with Professor Bojarski in Poland. So we screen at least 500 compound, and we identify a very interesting compound, cerodolin, which has a very strong affinity to the receptor. So as Carolina told you, we patented this molecule. And I will explain why we patented this molecule. So first, we identified this molecule. It has a very strong affinity for the receptor, but now we have to evaluate the effect of this molecule on the different pathway of the 5-HT7 receptor to know whether this molecule behaves as an agonist, antagonist, inverse agonist, or biased ligand. So to do that, so we evaluate we compare the effect of cerodolin with a reference agonist, the 5-CT, or reference antagonist SB269, and we use BRET assays. In fact, we use a BRET biosensor, which allow to detect the cyclic AMP level inside the cells. So as you can see here, when we evaluate the effect of 5-CT, so we observe an increase of the cyclic AMP response, as we expected, because 5-CT is an agonist. So SB is an inverse agonist, able to decrease the basal activity of the receptor, and we observe a decrease in cyclic AMP level, and cerodolin behaves as an inverse agonist. So we also tested cerodolin as an antagonist, and as you could see here, we detect and we see that the cerodolin does dependently decrease the cyclic AMP response induced by 5-CT. So in this slide, you can see that cerodolin behaves as an inverse antagonist on GS cyclic AMP pathway. And so to evaluate on different signaling pathway, we also evaluate the cerodolin on the ERK pathway. So as expected, when we tested the effect of 5-CT on this pathway, we observe a strong increase of ERK phosphorylation. But we were very surprised when we tested cerodolin on this same pathway, because we also observe an increase of ERK phosphorylation and a dose-dependent increase of ERK phosphorylation, meaning that cerodolin in this pathway acts as an agonist. So we confirm that this effect could be observed in the more relevant physiological systems. So we performed the same experiment on primary neuronal culture by immunohistochemistry, and you could see that cerodolin induces an increase of ERK phosphorylation in the same manner as 5-CT, the reference agonist. So we were very surprised because how molecule can increase ERK and decrease cyclic AMP. So we wanted to verify that the effect of cerodolin is very specific. And so to do that, we evaluate the effect of an antagonist, a known antagonist, the SB269. So we confirm that when we incubate the cells with the antagonist, the effect of the cerodolin is blocked. And in addition, when we perform the experiments on cells that do not express the receptor, we do not detect we didn't detect any increase of ERK phosphorylation in these cells, so confirming the specificity of our compound. So to conclude this part, so cerodolin behaves as an inverse agonist antagonist on one pathway, the cyclic AMP pathway, but it increases ERK phosphorylation on another pathway, and behaves as an agonist. So how a same molecule can inhibit one pathway and activate one pathway. So we speculate that in fact our cerodolin acts as a bias ligand, as I introduced you previously. So our cerodolin is able to inhibit G protein activation and to favor beta-arrestin activation. So we again use BRET assays to validate and to go deeper in the molecular mechanism of our compound, the cerodolin. So to evaluate the effect of the cerodolin on G protein signaling, so we perform BRET assay. We follow the G protein recruitment after activation of the receptor. So as you could see here, after incubation of the reference agonist, we could observe an increase of BRET signal. And as expected, because we observed before that cerodolin on this pathway act as an inverse agonist, it decreased the BRET signal. Cerodolin decreased like ASB, the BRET signal, acting as an inverse agonist. So by using BRET assays, you could also evaluate the recruitment of others, G protein, G12, GI, or GQ protein. So here it's just an illustration to show you how we could use BRET. And to go further, we decided to demonstrate that how cerodolin induce ERK phosphorylation through a G independent mechanism by inducing the recruitment of beta-arrestin. So again, we use BRET experiments and BRET assays to evaluate the effect of cerodolin on the recruitment of beta-arrestin. So we compare the effect of the cerodolin with 5-CT, the reference agonist, and we do not observe any beta-arrestin recruitment after 5-CT, but when we use cerodolin in increasing concentration, we could observe an increase of BRET signal. So we demonstrated by using beta-arrestin cow cell line that cerodolin and the effect on ERK phosphorylation is due to the beta-arrestin recruitment. So considering the agonist activity of our ligand and considering the role of the receptor, the 5-HT7 receptor, to control pain, we decided to evaluate the effect of cerodolin in different inflammatory pain models. Here you see the results that we obtain after injection of formalin in the paw of the animal. You can see that cerodolin decreases the paw licking time in the same extent as morphine. And interestingly, in another test, we tested the effect of cerodolin in another inflammatory pain model after injection of Fred adjuvant in the paw, and we measure the hypersensitivity of the animal with a von Frey filament. And I will not go too to go in the detail of the experiment, but when we injected the inflammatory agent, the CFA, we observed an hypersensitivity of the animal. And when we injected in the same time the serotonin, we reverse this hypersensitivity. And this effect is specific of the 5-HT7 receptor, because when we use mice deleted of the receptor, cow mice, 5-HT7 cow mice, we could not observe any effect of the serotonin. So to conclude, and I think it's my last slide, we identified the first arrestin bias ligand of this receptor. So we identify a new signaling pathway of the receptor involving beta-arrestin recruitment. We demonstrated the therapeutic potential of serotonin, which is able to reduce inflammatory but also chronic pain. And we speculate that this receptor can be an interesting opportunity to relieve pain in the future, because, as you know, neuropathic pain is considered as a public health problem, because over 15% of patients do not respond to current treatment. You know the problem of morphine and the dependence and tolerance due to the use of this kind of compounds, the opioid crisis, and the number of people who die every day in the United States. So identifying new therapeutic targets, such as why not 5-HT7 receptor, can be very an interesting opportunity in the future, and an alternative strategy to the use of opioid. So in conclusion, I hope that I convince you that the BRET is a powerful and a versatile tool to perform real-time measurement, to study in real-time protein-protein interaction. I hope that I convince you that NanoBRET has emerged as the most advanced system to perform experiment, and the GPCR signaling and drug screening remained key application of these BRET systems. So I want to thank all the members of my teams who participate to these studies. I want to thank all my collaborators who participate in these studies, and I want to thank the financial agency which support our research, and I want to thank you for your attention. So many thanks.